Good evening, everyone, and a warm welcome today for our UROWebinar entitled New Perspectives on Antibody-drug Conjugates as Targeted Therapy, from Basics to Clinical Practice. I have the honor to chair this session together with Professor Jeronimo, and today we will discuss about the role of these new therapies in the management of urological patients. This session is organized by the ESU in collaboration with the EAU section of Urological Research and Oncological Urology. And I'm sure it will be a very exciting and informative discussion on a rapidly evolving area in urological oncology. Today, we will explore the fundamentals of ADCs, examine key molecular targets, and discuss their growing role in clinical practice. And we will share insights into how these therapies could actually improve treatment precision and patient outcomes. With that, I'm pleased to hand over the word to Professor Jeronimo for the introduction of our speaker.
Thank you so much. Good afternoon or good evening, depending on the geography. So it is my great pleasure to introduce you Dr. Daniele Hagi. He is from Royal Marsden Hospital in London, and he is a genitourinary medical oncology. He's been working in the clinical research unit at that hospital, and he has a particular interest in molecular targets, including Nectin-4, as we know, very relevant for urological cancers, particularly to urothelial cancers. So the floor is yours, Daniel. Thank you so much for accepting the invitation to be here today.
Thank you. Good evening all. So thanks Carmen, thanks Giorgio for your kind invitation. So let me just share my screen. Okay, so let's start with this overview regarding the molecular targets of antibody-drug conjugates in urothelial malignancies. These are my disclosures. So let's start with the ADC evolution. So this is just an example of the TDM1. It's a quite old but approved ADC, so antibody-drug conjugate for breast cancer, so targeting the HER2. As you can see, the structure of the ADC was quite simple. So just including the antibody that can potentially, target a specific antigen on the surface of the cells. There's a linker, so basically linking the payload. The payload is basically a cytotoxic drug, usually, can be a specific chemotherapy with a very, very low dose of drug, causing the damage to the cell. After that, we are now managing the next generation ADCs. So basically increasing the drug antibody ratio. This kind of ADC have a higher potency, a slightly different mechanism of action. They are generally more cleavable. And moving into, the future direction that is now currently basically used within the clinical trials with new ADCs, increasing the numbers of potential targets, so antigens. So we have B or 3 specific antibody-drug conjugates, or by increasing the number of payloads, so potentially 2, 3, 4, or 5 payloads. So we can potentially give chemotherapy, we can give immunotherapy, we can give target therapy, or potentially radioligands as well. So very, very briefly, regarding the mechanism of action of the ADC. So as you can see, there's basically the target approach, so the binding to basically recognize the antigen on the surface of the cell. Then we have the internalization of the complex ADC antigen. We have a lysosomal degradation of the antibody portion of the drug. Then we have the release within the cell of the payload, and of course, the interaction with the target that can be DNA, can be topoisomerase, or definitely different targets. We have the death of the cell, and then the release of the payload within the extracellular matrix, and then eventually a bystander effect by killing the surrounding cells, so different off-target tumor cells. You know that the ADCs are definitely designed in order to expand the therapeutic windows. As you can see here, basically the chemotherapy has a minimal therapeutic window. By adding second, third, or fourth generation ADCs, we are expanding the efficacy of this drug by reducing the toxicity profile and try to avoid or spare what we call the off-target effect of these specific medications. We also can select specific payloads, so different drugs potentially based on different tumor types. We can select different target, as I told you before. So we have definitely a good drug for now. We also have prominent payload classes. So as you can see, as far as August 2024, last year, camptothecin and auristatin were the most used payloads within clinical development, so meaning clinical trials, and within FDA approved drugs. So let's start with the prostate cancer. So of course, as many of you already know, we have a lot of surface targets potentially expressed by the prostate cancer. Once again, more studied and expressed in the metastatic CRPC setting. As you can see here, I will not go into details, but definitely we have the PSMA, we have the B7H3, we have the STEAP1, we have the DLL3, we have the TROP2 expression, we have the CD46. So a lot of potential targets on the surface of the prostate cancer cells. And the therapeutic approach is based, as you can see, on the ADCs, because the ADCs are developing specific targets, specific medication, as long as potential CAR-T approaches or specific companions. But the ADCs are common within all these surface targets. Let's start with the PSMA targeting. So it's an old story, unfortunately, not successful. We have first clinical trials, including PSMA ADC monotherapy. This was a phase 2 trials, including more than 100 patients with a specific PSMA ADCs. Unfortunately, very few PSA responses. I mean, clinically meaningful responses, and 2 toxic deaths. So definitely not the right medication, not the right drug. We have another phase 1/2 clinical trials published as well, but unfortunately showing once again only very few responses and several toxicities. In this specific case, neurotoxicities was a dose-limiting toxic effect. So for the future, definitely the research is moving through bispecific T-cell engagers within clinical trials still ongoing, but targeting the PSMA expression. STEAP1. So STEAP1, once again, we had an ADC developed and basically studied in this phase 1 trial. Once again, few partial responses. As you can see, of the patients were achieving a partial response, slightly more than 10% achieving a PSA reduction, but once again, safety concerns. So several severe toxicities, so grade 1, grade 3, or grade 4. Once again, limited clinical activity, significant side effects, and so discontinuation of this ADC. But the research is moving. Once again, bispecific T-cell engagers are now under development and under study within clinical trials. TROP2 is a highly expressed transmembrane glycoprotein. It's involved in grown invasion and cell migration. We had this basket trial, including 11 patients, CRPC patients, unfortunately very, very few prostate cancer patients. Once again, overall response rate 9%. And so manageable toxicity profile in line with the sacituzumab govitecan, so the specific ADC against TROP2. And the research is moving once again by testing new ADC within clinical trials, such as the datopotamab, the deruxtecan, and newer one. CD46 is a membrane protein, basically protecting the cells against the complement-mediated cytotoxicity. Is highly expressed broadly in metastatic CRPC, either adenocarcinoma and neuroendocrine prostate cancer. Can be usefully used as tracer for the PET imaging as well. For the first time, we have a phase 1 first-in-human trial published recently, few weeks ago on JCO, studying basically the 446 an ADC against the CD46. As you can see here, for the first time, we can see in a highly heavy pretreated population an objective response rate of roughly 20% and a median duration of response more than 7 months. So definitely a step ahead. Delta-like ligand 3 is another target, is a Notch pathway inhibitor ligand. Is highly expressed on the surface of the poorly differentiated neuroendocrine tumors. Is highly expressed in the small cell lung cancer. It's also expressed in the neuroendocrine prostate cancer, detected in roughly 80% of the tumors. So we had a bad initial experience with the ROVA-T ADC. Is now discontinued because of several toxicity, so some concerns on the toxicity profile, and unfortunately a quite low response rate, roughly 13% of the patients just were responding. Tarlatamab is definitely a quite new approach. Is a DLL targeted T-cell engager, so it's not an ADC. Has been FDA approved last year, so in 2024, for the advanced small cell lung cancer. We have encouraging data recently shown at the ASCO last year annual meeting in DLL3 positive neuroendocrine prostate cancer, so advanced disease, showing a benefit, especially in positive and biomarker selected patients, showing also median duration of response at least quite encouraging. Once again, the research is moving, so including next generation ADCs, radioligand specific treatment, or new T-cell engagers as well. Last target is the B7H3 protein. Is part of the B7 family. Is involved in immune checkpoint molecules, so is highly expressed primary to the primary prostate cancer tumor, as well as the metastatic refractory, so CRPC setting. We have encouraging data by this phase 1 clinical trials testing the ifinatamab, the deruxtecan, is a specific B7H3 targeting ADC. was generally well tolerated, showing more than 30% of the response rate, with median duration of response roughly more than 4 months. We also have an ongoing clinical trial, the TARAMAC trial in metastatic CRPC, is led by Johan de Bono here at Marsden in London. It's basically testing the vobramitinib docarbazine, so it's another specific ADC. It is just a prostate-only clinical trial, but once again encouraging, efficacy data, but some concerns in terms of toxicity. There has been reported more than 4% of fatal treatment-related adverse events. Let's move to the relapsed or refractory germ cell tumors. CD30 is a cell surface protein. It's within the tumor necrosis factor receptor superfamily. Has been basically found within activated T cells and B cells. Is highly expressed in some kind of lymphomas, especially Hodgkin and non-Hodgkin lymphomas. And within the germ cell tumors is highly expressed in the embryonal carcinoma. We have a specific drug, so the brentuximab vedotin is an anti-CD30 antibody-drug conjugate. We have, unfortunately, just case series on that, 18 patients treated. As you can see, the positivity of the CD30 is highly correlated with a potential response in terms of overall response rates. These data were confirming a previous case series published by Andrea Necchi in germ cell tumors, so non-seminoma ones patients as well. The second and more definitely interesting and exciting target within the refractory germ cell tumors is the Claudin-6. Is a tight gap junction. It's highly expressed in the embryonal phase of the development and then lost. As you know, it's highly expressed by germ cell tumors. More than 90% of the tissues are expressing the Claudin-6, are positive. The mechanism of targeting is based on monoclonal antibodies, but unfortunately we have negative reports on that. ADCs, CAR T cell with or without vaccine, and B or 3 specific antibodies. These are the data derived from basket trials, recently presented at ESMO last year in 2024, including a lot of solid tumors. Within we have few cases of refractory germ cell tumors, encouraging response rates, encouraging tumor markers reductions as well. So I think that the direction is definitely now drawn, and we have some planned and ongoing phase 2 clinical trials, including ADC testing and CAR T cell with or without vaccine as well. Let's close with the urothelial carcinoma, so the most rich and interesting setting that we have for the ADC. So these are the key ADCs in the metastatic advanced urothelial cancer. Maybe you are just familiar with these names of medications, but the enfortumab vedotin is definitely quite famous nowadays. We have the sacituzumab govitecan, we have the trastuzumab deruxtecan, or the disitamab vedotin. As you can see here, we have different targets. So the enfortumab vedotin is targeting the Nectin-4. Sacituzumab is targeting TROP2, as said, and trastuzumab or disitamab are definitely targeting the HER2. And as you can see here in this table, we have some regulatory approvals as well by the FDA. All of them were roughly approved by the FDA or with the breakthrough designation. But unfortunately, the sacituzumab govitecan had withdrawn in 2024 based on the results of the phase 3 clinical trial. Let's go briefly on the ADC monotherapy history. So we are talking about very, very advanced disease, platinum refractory, immunotherapy refractory. This was the EV 301 clinical trial, basically without any selection of the Nectin-4 expression on the cells. Patients were randomized to receive enfortumab vedotin monotherapy versus second-line, third-line chemotherapy, so taxanes or vinflunin. The study was positive, and so we had the ongoing FDA approval registration for this drug. As well as, unfortunately, similar clinical trial, another phase 3 trial, the TROPiCS-04 trial testing sacituzumab, so a different target ADC. Same concept in terms of design of the clinical trial, randomizing patients to sacituzumab govitecan versus second/third-line chemotherapy. Unfortunately, the study has been presented by Petros Grivas at ESMO Asia last year, negative results, and then FDA approval withdrawn. Anti-HER2 setting. So we know that roughly 60 to 70% of the patients with advanced or localized urothelial cancer are expressing HER2. So this is a very, very good news for our patients, including if we are including the immunohistochemistry 1+ positivity, so a lot of patients. We have this trial, so the Destiny Pan-Tumor02 trial was a basket trial, including a lot of solid tumors, highly expressing HER2, so with immunohistochemistry positive for 2+ or 3+, so highly positive or very, very highly positive. Studying the trastuzumab deruxtecan monotherapy, the trial was positive. We got the FDA approval, sort of agnostic approval based on the HER2 positivity across all the tumor types. And regarding the bladder cancer, we have very, very encouraging data on the overall response rates, median PFS and median OS as well. This data has been confirmed by a similar trial. This was a combination of two phase 2 clinical trials from China, studying the disitamab vedotin, so similar medication but with different, payloads and slightly different antibody. Once again, including more than 100 patients highly expressing HER2 immunohistochemistry, with very, very, very promising results as well. We had, as I told you, we had the FDA breakthrough designation for this medication as well. So why combining ADC and immunotherapy? We have definitely some evidences, showing that we may obtain something. We may increase by adding an ADC to the immunotherapy. We may increase the efficacy. This is basically based on an increase of the immunogenic cell death. We have an increase in terms of dendritic cell maturation. We can potentiate basically the immunological memory. We can increase the activation of the T cell infiltration and the NK cells as well. And we can definitely modulate or increase the expression of the PD-L1. So potentially there's a quite strong, as if I may, quite strong rationale behind that. We have evidence that basically combining immunotherapy, so pembrolizumab to the enfortumab vedotin, is showing promising results. So this is the cohort A, the expansion cohort of the EV103 trial. So locally advanced or metastatic patient, cisplatin ineligible patients. So this is a first-line setting. We have recently an update of the results with a longer-term follow-up at ESMO 2024, showing that more than 40% of our patients are still alive treated with this combination. And we have another parallel cohort, the cohort K, of the same clinical trials, basically who randomize, the combination of the EV plus pembrolizumab versus the enfortumab vedotin alone, and showing definitely an improvement in terms of outcomes. What about the Nectin-4? So Nectin-4 is definitely a cell adhesion molecule. It's highly expressed on the most part of the urothelial cancer cells. It's approximately probably more than 90% or 95% of the tumors highly expressing Nectin-4, especially at the beginning or before any targeting treatment. We have very few data, especially retrospective data, on the correlation between Nectin-4 expression and responses to the enfortumab vedotin treatment. But we have the EV302 trial, so this was a first line up front setting, so a patient naive of any treatment with locally advanced or metastatic disease, receiving enfortumab vedotin plus pembrolizumab for the first time versus platinum-based chemotherapy, so cisplatin or carboplatin plus gemcitabine. Co-dual primary endpoint, progression-free survival by independent revision, and overall survival. This is the baseline characteristics of the population, quite well balanced. As you can see here, 30% of the patient in the experimental arm had an upper tract disease. So this is roughly quite new for metastatic setting clinical trials. Half of them, slightly less than half of the population was cisplatin ineligible, and 22% of the patient had liver mets. So quite badly selected population, if you want. These are the curves, so the results in terms of clearly a benefit in terms of progression-free survival as well as overall survival, nearly doubled the median overall survival, going from 16 months to more than 31 months as well. As you can see here, all the subgroup analysis, pre-specified analysis, showed basically an overall survival benefit. So including the cisplatin eligible or ineligible population, the highly expressing PD-L1 population or not, the presence or not of liver mets, the nodal-only disease as well, or irrespective of the Nectin-4 expression on the cells. So there's clearly no doubt that this is a pivotal achievement for the urothelial cancer, but we have some questions left. So which is the optimal treatment duration? What about the toxicities, costs, and definitely how to manage any kind of relapse after the EV pembro combination as first line. So we need definitely more time, we need more follow-up, and we need clinical trials on that. Alternatives of combining IO and ADCs. So we have some examples here with anti-HER2 ADCs. So trastuzumab deruxtecan or disitamab vedotin with or without immune checkpoint inhibitors, showing definitely probably a quite good benefit. And with the anti-TROP2 sacituzumab govitecan in refractory metastatic UC, showing more than 40% of the overall response rate and quite encouraging median overall survival as well. Several clinical trials are definitely ongoing on that. Very, very quickly, so we have a lot of proof of concept neoadjuvant trials, so MIBC. As you can see here, phase 1/single arm phase 2 trials targeting Nectin-4, targeting TROP2 or HER2, and showing definitely quite encouraging as well pathological complete response rates. Just the last phase 2 clinical trials with perioperative disitamab is basically, as you can see, including neoadjuvant and adjuvant phase compared to the other ongoing clinical trials. New ADC development, definitely yes. We have some new ADC targeting Nectin-4, HER2 as well, and TROP2, or the new appearance of the bispecific as well. The concept is definitely try to find probably a quite effective or same effective drug with less toxicities, and definitely, or a new medication potentially able to overcome acquired resistances. Some ongoing clinical trials in the metastatic setting as well, targeting HER2 or Nectin-4. Once again, disitamab vedotin is probably the most, commonly used anti-HER2 in the UC at the moment, with immunotherapy, so potentially pembro versus toripalimab, or an new anti-Nectin-4, the BT, in the DURAVELO 2 trial is a phase 2/3 trial, including patient upfront or previously treated, so 2 different cohorts. Once again, we have some data on the combinations of the ADC as well, so a dual ADC approach with or without immune checkpoint inhibitors. Moving back to the muscle invasive setting, we have 3 ongoing phase 3 clinical trials, so randomized clinical trials, potentially registrational clinical trials, all of them including EV, so enfortumab vedotin plus immunotherapy versus, the control arm. The Keynote 905 trial, so the second one in the middle, is basically enrolling patient cisplatin ineligible, as well as the Volga trial with 3 arms. So definitely we are waiting for the results of these clinical trials. Moving versus, the bladder sparing approach, we have some clinical trials testing the ADC alone or in combination with the radiotherapy in MIBC. There were some concerns regarding the safety and toxicity profile in this setting, but definitely there are still these trials ongoing. Just to close my overview on the ongoing clinical trials, let's move early, so to the non-muscle invasive setting. We have some trials, specifically these trials, the EV104 trials, basically with intravesical enfortumab vedotin, so with induction and maintenance phase in the high-risk BCG unresponsive non-muscle invasive bladder cancer, and 3 different trials in the BCG unexposed patients, so basically BCG naive patient. The first one is proposing the sacituzumab tirumotecan is a new different payload, but same concept, also with an anti-TROP2 ADC, and the last 2 clinical trials in selected patients, so with at least high expression of HER2, with disitamab vedotin plus BCG, and in the last one plus immunotherapy, so tislelizumab. So once again, definitely moving in the earlier phase of this setting. This is my last slide, just to try to summarize, so I will not go into details, but this is our landscape, our scenario in 2025 in terms of ADC in advanced urothelial carcinoma. As you can see here in blue light, you can see the Nectin-4 agents, so definitely the enfortumab vedotin and the newer ones. In green, you can find the anti-HER2, so as mentioned, the trastuzumab deruxtecan and disitamab vedotin, and in red or orange, you can find the sacituzumab govitecan. That is basically the older one and the newer one, so under study and within ongoing and promising clinical trials. I will left for later basically the chapter on the bispecific, but we have some approaching very exciting data on the bispecific as well. Thanks for your attention.
Thank you so much, Daniel, for this very comprehensive and very schematic way of presenting the new exciting data that we have been observed regarding usage of ADCs for urological cancers. And talking about that, and as you nicely showed, there are much more going on urothelial cancer than on the other urological malignancies. So can it be related to the fact that these the urothelial cancers are a bit more homogeneous regarding the targets, comparing with the others that tend to be more heterogeneous? Or is it a matter of, applicability of the other targets that are not so well studied in other tumors?
Yes, thanks for the question. So I think you're right, definitely. We have some, malignancies such as the prostate cancer with a long-term life expectancy. For example, a lot of approved medications. So we have the ARPI, we have the chemotherapy, 2 lines of chemo, we have the radioligands actually. So we have a lot of approved, for example, treatment for the prostate cancer. We know the metastatic CRPC is definitely quite aggressive. Is still a need, to find, but it's quite recent. The study of specific targets in this setting. Moving to the germ cell, fortunately, germ cells are mostly curable, so we have a cure rate that is very high with the first line chemotherapy. We have second line quite effective chemotherapy as well. So it's definitely a niche. Finding patients not responding, but fortunately, after years of failure of specific medications such as immunotherapy or target therapies, fortunately, we have a new target, the Claudin-6, is very promising. And so, as you said, yes. So I think we have specific targets for the UC, for the urothelial cancer, and so thanks to the first and positive trials assessing the EV efficacy, we are now living a new era.
And regarding the drug, the DAR, so the drug to antibody ratio, I know that there is at least some controversy because as far as I have been able to read, it can vary between 2 and 8, although 8 is a bit too pushy because it goes over the limit, and perhaps bispecific ADCs are indeed a matter of trying to reduce that DAR, so DAR, the drug to antibody ratio. So do you think that it's the way to go for using this specific in order to decrease that ratio that is of course associated with higher cytotoxicity at different levels? And you showed some examples. Can you comment on that?
Yeah, it's a very good question. So it's a hard answer. So definitely yes, I can say. So I think So definitely the right way to move is probably to try to test this kind of bispecific or trispecific ADCs. It's quite difficult to find, and the most important thing is to find a specific target for that, because potentially you can give 2 different drugs, so the approach is the same, because if you can find a specific target, for example, if the immunotherapy is working in a specific setting, you can just give a PD-L1 or PD-1 inhibitor and alongside a a specific antibody-drug conjugate, for example, for the Nectin-4. So this is the rationale of the combination of EV plus Pembro. But with, with a specific, just one bispecific, so one ADC probably is better in terms of trying to reduce the off-target effect, and so potentially try to spare toxicities. Yeah. So I think probably yes is the right way to go.
And as far as I saw in the several literature that I've read, and your presentation has shown that beautifully. So most of these approaches use cleavable linkers because they tend to be at least more friendly to use. But again, we know that this can lead to higher cytotoxicity because you need to have the acidic TMA, so all the environment required to make this prodrug really drug and effective. Can you comment on that? Why all the clinical trials and all the drug design goes to this cleavable linker instead of going for non-cleavable, which tend to be at least more stable when we are applying it systemically? At least that's my feeling. Do you want to comment on that?
Yeah, so basically it's in order, to try to reduce the off-target treatment, the off-target, side effects, especially on the normal cells. Potentially there's a bit of disadvantage, by using a more cleavable ADC, but I think that this is the current direction. So definitely it's better to try to use them, in order to spare the off-target toxicities. Yeah, I think definitely, this is a matter of discussion. So definitely recently, because if you think about this sacituzumab govitecan, so we had the failure of a very, very huge phase 3 clinical trial. So probably because of some, issues regarding the design of the study, or maybe, the management of the expected or unexpected toxicity, for example. So by changing the payload could be probably another way to go, because, the target is definitely a good target, because if you think about the sacituzumab based treatment are now, we have yesterday the press release of the breast cancer. So we have positive results regarding the ADC, especially, the sacituzumab as well in first line for the breast cancer. So I think definitely there is nothing, against the sacituzumab govitecan itself. Is definitely more, our way to study, to learn about the toxicity, how to manage them, or how to prevent them. Definitely, yes. More than, the specific characteristic of the single drug.
Mm-hmm. I don't know if you are familiar, but when we are talking about preclinical studies, there are some studies now approaching using PROTACs, to be able to increase the efficacy. So the proteolysis targeting chimeras that are more, well, at least in vitro, they tend to be more specific and effective when adding or getting the drug inside and being internalized. Do you have any experience, or do you know if there is mechanistically a way of to push with these PROTACs instead of the conventional ADCs?
No, unfortunately, I cannot comment on that.
Okay. Yeah, because
sorry.
Yeah, because there are some studies using this approach in order to increase the efficacy of some ADCs. Okay, so meanwhile, we don't have any question from Q&A, so I invite those of
We
you
actually
that
have, sorry, Carmen, we have a couple of questions actually.
We do? I cannot see them. I don't know. Okay, so please, can you?
I will take the honor to read the questions. So
Okay.
Gigi is asking us about the cost and the economic considerations which are likely to influence the inclusion of ADC therapies in urological malignancy treatment pathway.
Yeah, it's another very, very good question. So the costs, unfortunately, for now are very, very high. So in terms of, you know, for example, I'm working and living here in the UK, in the United Kingdom. We don't have the approval yet from the NICE committee of the enfortumab vedotin. Either in subsequent lines, and you know, we are waiting for the approval as first line setting in combination with pembro. So definitely costs matter. So it's a very, very, you know, a tricky discussion with the local authorities, you know, in terms of we need probably to make a huge effort, you know, in order to make the sustainability of this combination of drugs, especially if we are not selecting patient by using a biomarker. So this could be an issue, especially for the enfortumab vedotin, because it's not required by the regulatory agencies. If we are going to offer, you know, a broad indication of the first line, for example, so including cis eligible or cis ineligible patients as well. So definitely would be an issue for the future. Yes, I agree.
And the second question we have is related to the options for renal cell carcinoma, penile cancer. Are you aware of studies in this setting which can actually, say, be key for the management of these diseases in selected settings, actually?
Yeah, definitely, yes. There are some ongoing clinical trials in RCC. But you know, it's very, very difficult, you know, because it's a completely different disease. So it's basically an angiogenic driven or immunogenic driven disease. So we have definitely less, I think, in my opinion, less options in terms of ADC in this specific subset. But we'll see definitely in the future. Regarding the rare cancers such as the penile, definitely I think could be, there are some clinical trials using, for example, EV in the same context, so treating patients across you know, all the GU malignancies, so including potentially rare tumors such as the germ cells or the penile cancer as well. We need to find the specific biomarkers. So some of the trials are including patients with high expression of Nectin-4, for example. But you know, is the same issue, and including all the rare cancer. So it's very, very difficult to find numbers. It's very, very difficult to find patients. It's very, very difficult to work all together. So I mean, including a worldwide action. So so definitely yes, it's a huge effort, but definitely it's worth.
Thank you. And I have a question for you actually about prostate cancer. So besides PSA, you did a great presentation and gave us a lot of potential targets. But at the end of the day, urologists are very simple-minded and we always think about PSMA. So do you think this is something that will be somehow feasible in the next future? We know about lutetium PSMA. Is this something that can replace theragnostic? And do you see a role for this? Because when we talk about theragnostics, it's also appealing that we have imaging at the same time. So that's why PSMA, in my mind, remains for urologists an attractive target for also ADCs.
Yeah, right. So yes, I think so. I think the answer is yes. We have, as I told you, we have very, very quite old data with basically not showing a good response in terms of the old ADC monotherapy against the PSMA. We now have the bispecific and trispecific ADCs, so potentially targeting the radioligands as well, so basically linked to the antibody. So definitely, yes, would be the future. So the main issue with the PSMA is that is, you know, is highly expressed, so in some specific tissues. But definitely, yes, I think the future would include definitely the ADC as well. So there are ongoing trials with new ADCs with B-specific, anti-specific engagers. So definitely, yes.
So thank you very much. I don't know, Carmen, if you have other questions or if you wanna.
Well, meanwhile, I was remember one thing. Well, I'm sorry, but I'm basic translational researcher, and I just came to my mind when you start to talk, when you mentioned the targets, and of course it's important to have targets. But one issue that really puzzles me is that we know that when the tumors are submitted to different therapies, they tend to be more genomic unstable. And we know that it's relevant when we are using as targets and when we are using as payloads, drugs that really goes to DNA breakage, or to really target DNA in order to induce apoptosis and therefore to have its effect. Do you have any experience on that? So tumors that are suffered different treatments, and at the end they do not respond or they become resistant to other kinds of treatments, they should be able to respond better to this approach. What is your experience, or what you know about that? Can you comment? Is it too silly to think on this?
No, absolutely not. Absolutely not. You mean with a specific ADC therapy? Yeah. So okay. Mm-hmm. So I think it depends on the target. So it's very, very difficult to tell you exactly which kind of tumor is expressing or a specific target. So it's not so easy, it's very, very probably, tumor specific or payload specific as well. Mm-hmm. But definitely, if you think about the breast, because we are all learning about the breast cancer, for example, in terms of ADC, anti-HER2 ADCs. Mm-hmm. We have nowadays, the data on the non-expressing target tumors. So potentially, if you think about the low HER2 breast tumors or ultra low HER2 breast tumors, they are still responding to the HER2 directed therapies. So probably we are missing something there. So I'm not sure if you are not seeing, for example, specific expression of the HER2 in the specific context, because we are using different criteria in terms of selecting patient expressing HER2, for example, NGS versus immunohistochemistry, or because this kind of base standard effects. So basically, you just need to target this very, very small proportion of the cell just to get a sort of cascade effect, so something huge or huger than expected. So I'm not sure definitely if the most important thing is related to the antigen or the cell or the payload. So I think it's too early to give you a specific answer on that.
Mm-hmm. Thank you so much for this very, very comprehensive and very informative talk on this relevant issue and very hot topic. So it's everything from my side. Thank you again for being here. And I don't know, Giorgio, if you want to close up and say something.
I want to thank Daniela and Carmen for this great discussion. Personally, I learned a lot. So I think this is a very, very hot topic for urologists, and we need to be ready for what is coming from our colleagues in terms of these new therapies in the management of many urological diseases. So thank you again, Daniela. Thank you, Carmen. It was a great evening, and I hope you have a nice evening now.
Thank you so much. Bye-bye. Take care.
Bye.